mission to engage students in local environmental science projects. I would like to thank everyone who is signed into this Zoom webinar. It's just one quick organizational detail before I introduce our webinar speaker. If you wish to ask questions, please use the questions box or the chat box in your panel and type your query. At the end of the presentation, we will read your question to the presenter, or the presenter will rather pick it, and he will answer the question. So now I would like to introduce our webinar speaker. Morgan McCarthy is a PhD fellow at the Marie Sklodowska-Curie Talent Doctoral Program at the University of Copenhagen, where he's working on in the marine mammal group. Originally from Cohasset here in Massachusetts, Morgan completed a Bachelor of Arts in the Earth and Planetary Sciences of the Johns Hopkins University and a joint Erasmus Mundus Master of Science in Tropical Biodiversity and Ecosystems at the Universities of Brussels and Belgium, Florence, Italy, and Queensland, Australia. After graduating from his master's thesis investigating a dugong population collapse in southeast Queensland, Australia, using DNA extracted from museum collection bones, Morgan interned at NOAA's Southwest Fisheries Science Center at the Marine Mammal Genetics Program in San Diego, California, where he worked on assembling whole genomes of beaked whales. This research campaign kicked off in Guadalupe, Mexico, where he collected samples which were used to better understand island population dynamics in beaked whales. Presently, Morgan resides in Copenhagen, Denmark, and has continued to work on aspects of beaked whale ecology and evolution, and also studies in an extinct population of gray whales in the Atlantic Ocean using ancient DNA, and collaborated on a study using just water samples to extract DNA from bowhead whales in Greenland. So the title of Morgan's presentation is Big Data for Big Mammals, How Genomics Can Be Used to Study the Ecology and Evolution of Whales. Morgan, the virtual floor is yours.
Thank you, Carson, for the introduction. I'll just share my screen here. Okay. So I just want to say thank you for everyone. Thank you to everyone for attending. It's great to see so many familiar faces and to see that so many people are interested in what I've been working on. And I'm going to open it up by going through a little bit of my path from a researcher at CSCR to where I am now as a PhD student, still doing research. And then I'm going to talk a little bit about the themes of my research and in general, how one can apply genomic data to studying the ecology and evolution of marine mammals. And I'm mainly going to focus on whales today. So let's get started. So I began my research experience here in Cohasset. Actually, my first field site I can see from where I'm sitting right now. It was a wetland in Little Harbor, and what we were interested in looking at was baseline measurements for water quality, plant biodiversity, and fish biodiversity over time. And that's Jackie Chase there on the left, who was my the team leader. And this is where I started to develop my skills in research. Now, after I left Cohasset High School, I went to the Johns Hopkins University, and there I studied a Bachelor's of Arts in Earth and Planetary Sciences. And in my junior year summer, I had the opportunity to go and work at a research group in Germany at the University of Bremen in conjunction with the Alfred Wegener Institute for Marine and Polar Research. And I worked with Simon Jungblut here on the top right, who was a PhD student himself at the time. And we were living on Helgoland for 3-week periods of a time. And we were studying the dietary preferences of different species of crabs and trying to assess whether or not they were competing for resources. And we were using Helgoland as our island system. And that was really where I sort of solidified my passion for research in marine science. And so after I graduated from Johns Hopkins, I decided to begin a master's with focus more in marine biology. And I did that through an international Erasmus Mundus degree in tropical biodiversity and ecosystems. And while I got a general background in tropical biology, I focused on the marine aspects. And it was here that through a consortium of schools that are organized by the University of Brussels, I started my first semester at the University of Florence, had a second semester at the University of Queensland, where I would eventually meet my master's thesis supervisor, and then had my third semester at the University of Brussels. And in my final semester, I went back to Queensland, Australia, and worked on a marine mammal called the dugong. And dugongs are here. Here's a close-up photo. And they are the closest living relative to them today is the manatee, which many of you may be familiar with down in Florida. And dugongs are fully oceanic and live in the Pacific, on the coast of Australia, all the way even to Eastern Africa. And I began not only research on dugongs, but I had my first experience working with historical or ancient DNA. And my research question for my master's was if using skulls from a museum collection could inform us about what the health of the Queensland dugong population was like before and after a government shark net program was implemented. So in the early and mid 1900s, tourism became very popular in Australia, and the government started to install shark nets to prevent sharks from getting too close to beaches and having interactions with swimmers. But unfortunately, some dugongs, sea turtles, and dolphins were caught in these nets, and they actually drowned. And some aerial survey estimates projected that up to 98% of the population had been decimated by these and other factors such as tropical storms. And so I was interested in looking at whether or not the dugongs that had survived in central Queensland today, if they had lost a significant portion of their genetic variation. And the way I was able to have an insight into this was to actually go to the museums and look at the bones from the dugongs that died during the shark netting program. But I'll talk more about these strategies in ancient DNA later on. So after I completed, so in Australia, not only did I have the chance to work on ancient DNA in dugongs, but I also got to work in the field and had some really interesting and fun experiences working with Janet Lanyon at the University of Queensland. And she is the leader of the Marine Vertebrate Ecology Research Group. And she has, for over 10 years now, been running a dugong health assessment where she monitors dugong health in Moreton Bay, and under the supervision and assistance of veterinarians and other marine biologists, is able to take samples of dugongs even by bringing them on board a research vessel and taking blood measurements and skin samples for genetics analysis and taking CO2 measurements. And some of the work is also able to be conducted in the water. And so I made a video to share with you what some experiences as a marine biologist I have had are. So I'll just play this, and it's a little long, so I'm going to skip to 30 seconds in. So that's just to demonstrate some of the field work that I helped carry out, and to highlight some of the techniques we were taking: body measurements, fluke measurements, looking for what sort of food was being consumed. And a lot of times people ask if the scars on the back of those dugongs come from boat strikes. And while some of them do, they're much more distinct, but those were actually just from tusks that erupt from older dugongs. When they are sort of jostling for mating position, they have these rake marks that they leave behind on other dugongs. So that's kind of where those scratches come from. And after I completed my master's, I went to intern with the National Oceanic and Atmospheric Administration under Phil Moran, who today continues to be a co-supervisor for my PhD. And it was at NOAA's Southwest Fisheries Science Center that I was really introduced first to beaked whales. And beaked whales are a group of understudied whales. There's about 23, but there's probably upwards of 26 species of them. And they live far offshore and dive into underwater canyons to feed on squid and fish. And we don't know a lot about them because since they're so far off the continental shelf, they're really hard to observe. So you either need to be sort of along an island chain or take a boat out, sometimes 2 hours just to observe them. And because they spend so much time at depth, when they surface, it's perhaps for only 2 minutes or so before they dive back down for anywhere between 15 minutes to 2 hours and 45 minutes. So depending on what sort of dive cycle they're on, they could reappear half a mile away from where you're looking for them. And so it was at NOAA that I first began developing skills in bioinformatics and learning how to assemble genomes, and developing the skills that I use today in my PhD. But while I was in San Diego, I also had the opportunity to work for a scuba/shark diving company called ST Expeditions. And here's a picture of me on one of the scuba expeditions with a California sea lion. And that was a really interesting insight into what working in ecotourism was like. And finally, just so people know who are pursuing marine biology, it's not always rosy. I was also a dishwasher at the same time, and that was just to make ends meet. But important for prospective students to know that sometimes you have to make some sacrifices to keep pursuing your passion. And it was towards the end of my internship and at the start of my graduate work that I was able to take part on an expedition to Guadalupe Island to study Cuvier's beaked whale. Now, Cuvier's beaked whale is in the illustration here, and this is the organization that's based in Ensenada, Mexico, who are led by Gustavo, and they study this population of beaked whales that live in Guadalupe Island. Now, Guadalupe, some of you might know the name because of Shark Week, and it's a great white shark potential breeding ground. And it's also home to the northern elephant seal. And a few decades ago, the northern elephant seal was actually declared extinct twice, but individuals that survived on this island were able to recolonate the west coast in California. And now there's over tens of thousands of northern elephant seals. So here's another video demonstrating some of the techniques that we were using to study Cuvier's beaked whale, from photo IDs to add to an existing catalog to try to track who's coming back over the years to the same island, but also to compare the photo catalog at Guadalupe Island to other islands in Southern California to try to figure out how far exactly do individuals travel in the species. And we also deployed some acoustic monitors, you'll see. And there are some marine mammalogists where I focus mainly on genetics. There are some people who specialize in acoustic sounds, and they can say things about population size or species ID just by listening to species. So here's another fieldwork video I have to share with you. So that whale you saw in the final image, that's Cuvier's beaked whale, and it holds the world's recorded record, at least for the deepest and longest dive, just under 3,000 meters and 3 hours, just about. So yeah, a really interesting species, and the samples we collected there will be used in future genetic analysis and ongoing projects by Gustavo and his team. And so that takes me up to where I am now, which is at the evolutionary genomics section at the University of Copenhagen. And while I've shown a lot of cool videos about fieldwork, me at this desk is where I actually spend most of my time crunching numbers and writing up results. And so I currently work under Morten Tunge Olsen here, and I'm co-supervised by Phil Morin from NOAA. And we are located within the Globe Institute, now in the Faculty of Health and Medical Sciences. In general, the lab focuses on the themes of environmental DNA and how you can use it to monitor marine mammals, as well as speciation genomics and where one population becomes distinct from another. And we also work in ancient DNA. And I'm going to talk to you about those themes as we move along in the presentation. So overall, like I was saying, my research directions are in marine mammal molecular ecology and in speciation genomics. And so you're probably wondering, what am I even talking about? And so what I'm looking at in molecular ecology is, what can DNA tell us about the evolution, species interactions, ancestry, population movements of marine mammals? And so sometimes it feels like I'm a forensic scientist, but just for marine mammals, and I'm seeing what we can learn from the DNA. And the other major research theme I focus on is in speciation genomics. So what does speciation look like in a genome? And there's different schools of thoughts about species concept, and some people don't think a species exists at all. But the bottom line is, in order to be able to conserve something, you need to be able to identify either ecological significant units or various species. And so I'm trying to quantify what a speciation looks like in a genome. So I'm going to talk to you about a few different projects and how we can apply big data to answering our research questions. And the first is a project I'm working on that's looking at the extinct Atlantic gray whale and looking at the relationship to extant Pacific whales, or whales that still are living today. So on this map here, you can see the red portion shows where gray whales used to live. So on the eastern coast of the United States, there were populations. We also know that they lived in the Mediterranean, all the way up through the North Sea and into Scandinavia. And we knew they lived around Iceland as well. And gray whale populations today live on the west coast of the United States and Mexico, and as well as on the east coast of Japan, China, and Russia. And it's thought that gray whales went extinct from a combination of factors, most likely the impact of whaling, but probably more so having to do with climate change in their habitat. But this is an active research question, something I'm interested in. And I'm wondering whether or not the extinct Atlantic gray whales were actually a separate species or not. Because it's possible we lost an entire species in the Atlantic, or potentially it was just a subpopulation or some individuals that had moved from the Pacific into the Atlantic. And I try to tackle this question using gray whale DNA. And so I'm going to go briefly into some genetics terms and what molecular markers we can use, but I'm trying to boil it down so that everyone can understand, and you don't need to be in a genomics lab to deal with it. And so basically what I'm talking about in this project is something that's called your mitochondrial DNA. And so everyone has a blueprint or their genome for how they're built. but actually inside your cells, there's an organelle, the mitochondrion, and it has its own DNA, and that's the mitochondrial DNA. And there's an interesting property about mitochondrial DNA, and that's it's passed down maternally. So your mitochondrial DNA is the same as your mother's, and all of your biological siblings share the same mitochondrial DNA. And we can use this to tackle the question about gray whale relationships. And I'm just going to demonstrate how this can be used. And that's, let's say you have 4 siblings, you all share mitochondrial DNA, and one of you moves to Japan. And in 200 years, researchers are finding human bones and they're sequencing the DNA, and they find that your mitochondrial DNA matches your 4 siblings who happen to stay, in my case, say Cohasset. And so you can use that information that there is a mitochondrial DNA matching that of a larger population Cohasset to infer that at some point, someone must have moved from Cohasset to Japan. And the same way that you can infer that with human DNA, I'm applying that to gray whales to try to infer how closely related the Pacific and Atlantic populations are. And this is actually a picture of a mitochondrial DNA, and it is, or the whole genome, and it's actually in a loop form, and it's about 16,500 base pairs. And when I'm talking about a base pair, I mean these little letters that you see. So these letters ATGC code for the blueprint of your whole body, as well as the mitochondrial genome. And when I'm looking at a comparison, these two, number 1 and 2, and these are sample names, those are both gray whale reads from this 16,500 base pair loop. I've basically just zoomed in and I screenshotted maybe 50 base pairs. And what I'm looking for is, notice how most of these match up, but it gets to a certain point where there's a CT that don't line up. And what I'm trying to do is quantify the number of differences between 2 different sequences. And if things are more related or had a common ancestor more closely in the past, there's going to be more of these that line up. And if they're more distantly related, there'll be a higher number of mutations that have just accumulated naturally through time. But just to put this into perspective, I lined up the modern human mitochondrial genome with a Neanderthal genome, and they have 98.7% genetic identity. So if you think about what's the difference in a species on the genomic level, think about 98.7 of these characters actually being shared between a modern human and a Neanderthal. So you're probably wondering, well, how do you extract DNA from a species or subpopulation that's already extinct? And the answer to this is through museum collections primarily. Now, here's a picture of me at the Zoological Museum in Copenhagen. And while there's some whales on display, in the back rooms and the storage closets, there are hallways with whale bones, all sorts of bones. And museums are a really great resource for researchers who are trying to look at populations that are difficult to actually sample. For instance, with beaked whales, it's hard to find them in the field, but they end up in museum collections, and then you can easily sample a number of species just from one collection room. Whale bones can be found in archaeological dig sites. So people find bones all the time. They could have been from potential past hunts, or just a whale that was beached onto a beach, and then over time sediment filled in, and now they're finding that hundreds or thousands of years later. And even here, this is a picture of an old commercial whaling station in Svalbard, Norway. And these are not northern bottlenose whale skulls that have just been deposited on the beach. Now, while every bone sample could be a candidate for DNA, it doesn't mean you'll find viable DNA to answer your question in each sample. DNA breaks down basically once an organism dies, and depending on light conditions, so sun exposure or temperatures and humidity, it can last longer, or it can degrade very quickly. And so I continued the work that was begun by Camilla Speller, who's a professor now at the University of British Columbia. But she began this project by contacting museums and collaborators and collecting all of the Atlantic and Pacific gray whale bones that she could find. and she began the extraction process. Now, I'm just going to go briefly through the schematic to try to inform you on what extraction looks like. And basically, you have your study organism. In this case, it's the woolly mammoth. And you have your sample. And this could be bones, teeth, ancient hair follicles that were found. And this gets ground down. And basically, chemicals are added to it in the laboratory. And inside of this chemical solution with your ground material, you send in primers, and these primers are like detectives. They know what DNA fragments they're looking for. And while we can't look at a solution with crushed bone and know if the genome or genetic material we're looking for is there, the primers go in on a molecular level, they find these sites, and then they can amplify them through a process called PCR. It's the polymerase chain reaction. And that's a term many of you might have become familiar with recently through COVID-19. So if you actually get a PCR test, it's most likely that a lab technician or a researcher is taking your mucosal or your saliva sample, and they're actually telling biomolecules to go in and try to amplify the COVID-19 genome. And if it doesn't find the genome, there's nothing to amplify, then your test will come back negative. But if it does find some of the genome and it amplifies it to a level where we can recognize it, then you know you're positive. So in the same way that people get tested for COVID-19, we can use the power of PCR and amplify regions of the genome to answer our research question. So in short, that's what we do until we have enough fragments that we can then assemble them into the whole genome. And some interesting preliminary results from this study is that, one, we were able to extract DNA from a 39,000-year-old gray whale, which is cool because it gives us some insight into what was going on way far back in history. But also the sequence data suggests that the Atlantic population has actually had multiple migration events from the Pacific Ocean into the Atlantic. Potentially around 85,000 years ago was one migration, and more recently 10 to 15,000 years ago. Now, some of you are probably wondering how this happens and how one dates these types of events. And just like if you know you can walk 1 mile in 15 minutes or 18 minutes, and I would ask you how long do you think it's going to take to walk 5 miles, you can scale it up by knowing your pace. And the same thing can happen with DNA, whereas if you can estimate how long it takes a genome to mutate, you can count the number of mutations between different genomes, and then you can scale it up by the mutation rate and try to make an inferred estimate on how many thousands or millions of years ago two sequences were actually had a common ancestor. And how could Atlantic gray whales have come from the Pacific can be looked at and correlating our time estimates with climate fluctuations. So this is a photo of what the Earth looked like before or just at the last glacial maximum. And basically when we had colder conditions, the sea level was lower because more water was locked up into glaciers. And so if you can see here on the top of the map, there wouldn't have been a path for anything to cross from the Atlantic to the Pacific. But as the Earth started to warm 10 to 15,000 years ago, those ice blocks began to melt and things were able to make movements from the Pacific to the Atlantic. And so that's right now what other data from other people who have studied this research question have suggested, and what our results are also suggesting. And it's interesting because just in the past 10 years or so, there have been sporadic sightings of this extinct species in the Atlantic, off the coast of Spain, off the coast of Namibia. And so it's become a question of, were these individuals that were remnant of the Atlantic population? And the consensus is that they probably just were able to make the travel from the Pacific into the Atlantic. And another theme I want to talk to you about is environmental DNA. And this is a study that just came out of my lab group that was led by Dora and Natasha and Morton. And it was actually using water samples to try to gather some of the same genetic information that you can get from a skin sample. So just to explain a bit further, usually when you get a skin sample from a whale, you either find an individual stranded, and then you can cut out a piece of the skin after they've died. Or what happens is in the field is usually you fire a modified projectile from either a crossbow or a rifle. And what happens is there's a sharp edge and a rubber stopper that floats. And so it usually enters the blubber and bounces off and then is floating in the water, and then you can scoop it up using your field boat. But what eDNA or environmental DNA looks to target is actually DNA that's floating freely in the water column. So there's DNA all around us. We shed our skin, the DNA from our lunch ends up on our surfaces, in our dining room table. And in the same way, whale DNA or fish DNA is floating around in the water, because as the whale swims around, skin cells are shed, it excretes excrement. Through whale blows, some mucosal cells come out and land in the water. And what you can try to do is collect the water sample containing DNA in a beaker or any sort of glass. Or what's being employed as well, not in this particular study, is actually flying drones through whale blows. And so these drones will fly through a whale blow with a petri dish connected to the bottom. And then researchers will try to extract all the DNA that was found on the petri dish. And so this environmental DNA is sort of the cutting edge now in marine mammal biodiversity monitoring, but in other biodiversity monitoring, because it's less invasive. You don't actually have to interfere with the whale to get the sample, but it also could be used to detect cryptic species. So species that are really difficult to find in the wild, like beaked whales, or species that you might not know are there, or they're so close to extinction that it's hard to visually see them. If instead you could just take a water sample to try and detect if they're there or not, that would save a lot of effort as well as cost in monitoring biodiversity. And the overall workflow for environmental DNA is you take your ecosystem or your study site, you sample the water in any sort of tube, you filter the water through a filter, and similarly to the ancient DNA, it goes to the lab, the DNA is extracted, you amplify all the DNA that exists in the sample, and then you can match up the sequences you find in a database with known genetics, known genetic reads. So if somebody had another study and they looked at bowhead whales, they have deposited the sequencing reads into the database. And then when you take your sequencing reads and go to the database, you can actually match them up with existing bowhead whales. And so not only do you target, or not only can you target your study organism, but you can gather information about everything that's living in the water sample or everything that passed through that water sample. And so in this way, you can see there's just 2% of the actual reads that were sequenced corresponded to any sort of whale. So there's still a lot of information in here that people can use to now study and sort of what's the community composition of water samples in Greenland, and then they can track that over time. So this is part of ongoing work that Dora's focusing on, and I helped her with the bioinformatics. But through the study, we're able to show that environmental DNA can represent overall genetic clusters that have been identified through traditional skin sampling and then population genetics. So you can do similar studies using just water samples, but this technique has a long way to go in biodiversity monitoring. And finally, I want to talk to you a little bit about genomics and speciation genomics, which is the third area I'm interested in. Now, just so you can comprehend sort of the big data behind genomics, I just want to share with you that in the human genome, there are about 3 billion nucleotides, or those letters I showed you earlier. And written out, the entire human genome would stretch 5,592 miles and would fill 1 million pages, 5,000 books stacked 200 feet high, or 200 telephone directories. So it's just to demonstrate exactly how much information is contained in the human genome, or any genome really. So for a marine mammal, the genome can be anywhere between 2 to 3 billion nucleotides, or those letters I showed. So it's on a similar order to the human genome. But just from one genome, you can try to estimate things like inbreeding, population size over time, how well populations are connected, and if there have been any population changes in the past. For example, just from a single genome in a woolly mammoth, you can see that there was a population collapse as global temperatures started to rise around 10,000, 15,000 years ago. And so when I actually look at a genome, this is what I'm looking at. There's a text file with millions, billions of these characters, and it's all coding for something. And sometimes you know what you're looking at, but other times the genome hasn't been annotated and it could be correspond to anything. But the point is there's an exhausting amount of data in the genome for a single human to look through. And so you can use computing resources to try to filter through all of the genome and pull out the parts that are informative to your study question or what you're looking at. So just for example, if you had a 64 gigabyte iPhone, you'd be able to hold 23 genomes on it before you ran out of storage. So it's really computationally intense and takes up a lot of space. But what I'm using whale genomes for is to look at speciation. And in 2016, there was a whale that washed ashore on the west coast of the US. And researchers were called to the site and asked what the species was. And for a long time, researchers have thought that these two whales here that I'm circling, Baird's beaked whale and Sato's beaked whale, were actually the same species. They both live in the northern hemisphere, have been seen around Alaska and Japan, and looked similar, except Sato's beaked whale was darker in color and smaller, whereas Baird's beaked whale was larger and more gray in color. But just like in nature, sometimes a golden retriever looks different than a black lab, but they're both dogs. This can happen to wild species as well. And so researchers were starting to investigate whether or not this, the black form of Baird's beaked whale was actually a separate species. And when I went to San Diego, that was what I undertook, but on the genome level, because previous work in the mitochondrial genome had suggested that they were actually different species. And since I began this work, a Japanese team led by Yamada et al. Was able to collect enough skulls from museums that were misidentified as Baird's beaked whale and make an official species designation for it. So I'm still pursuing my questions of how does speciation look on the genomic level. And what we've found is that the northern and southern hemisphere relatives are actually more genetically similar than they are to the smaller whale living in the northern hemisphere as well. Now, this is a bit unexpected because these two both lived in the northern hemisphere, but at the same time, they look fairly similar. So you might be thinking it actually looks more clear to us, but it's not always the case. And so of the genetic identity, they're actually 99.81% identical, which is less than the Sato's beaked whale, which is 99.62% identical. But when you're looking at billions of base pairs, that's over 3 million nucleotides that are different. And so using the whole genomes of all three species, I'm able to try to place them in the larger sort of tree of life for whales. And so that's something that's ongoing, and I'm writing that up with collaborators now. And so future work for me and in genomics is what sort of range does Sato's beaked whale have and how many are there? Is the population at risk because it is so rare, or is it just hard to find? And what factors drive the speciation of sister species that have a northern southern hemisphere split? So I'm interested in species like the northern elephant seal and southern elephant seal, or the southern right whale and the North Atlantic right whale, because you have species that are very similar, but somehow the ancestral population was split and they now today ended up on opposite sides of the equator. And so I'm in general interested in what factors drive that sort of separation. And here are some of my works cited. And I just want to say thank you to everyone. I've been really lucky over the years to have a number of mentors and supervisors who have helped me move forward in the field and taken a chance on me. So thank you to all my supervisors, Phil and Morton and Janet and Jenny. And thank you to Carson and Jack for inviting me to speak here today. And of course to family, friends, and lab mates who are a lot of fun along the way. So any questions?
Very much, Morgan. That was awesome. So was one question here from Anne Tomei. Morgan, want to read it yourself and answer it?
Let me pull up the chat.
I can read that for you. That's what genome percentage difference will occur in order to classify organisms as being in a different species?
So great question. And if I had a very straightforward answer for you, there would be a lot less debate in the genomics community. But basically, it's interesting because for whales, species were usually defined based on skull morphology. So if you could collect enough skulls from an individual, you could take measurements and say, this is significantly different from this species, so we're going to call this a new species. And they live in different ocean basins, and it doesn't look like they breed together anymore. But whale biologists needed to sort of come to a consensus, because for some of these species, they're so elusive, and some of them we haven't even observed live in the wild, where they had to say, if we can get a genetic sample, are there any cutoffs we can give to try to define a species? And so in general, if you look at a particular portion of the mitochondrial genome, if they're about less than 98% similar, they're probably different species. But it could be a subspecies that's just been isolated from the other population. But there's a lot of debate about this now. And what can complicate this, especially on the genome, is, this is what I'm finding for the northern and southern beaked whales I'm looking at, is that they had an initial divergence, but at some point, an individual moved back either to the northern or southern hemisphere, had a breeding event, and then moved back. So you can actually have genomes that look like they're more similar, even though most of the population could actually be very split. So it's an excellent question, and I don't know if the more genomic information we have might just make that even more difficult question to answer. But from a conservation perspective, managers sort of at times need a hard number to rely on in order to protect species. So yeah, that's that.
And I think John Leffel has a comment.
Hi. Hey, Morgan, great presentation. This is fantastic. I'm a liberal arts major, and I think you made it simple enough so that even I can understand it. So thank you.
Thanks.
Really good stuff. Hey, just a question for you, though. You've looked at some very, very long periods of time in the clinical and data sense about weather patterns, which made me wonder, what do you see in terms of the climate change pattern? I mean, are we within bounds, or are we really stepping out of ranges that you've seen over your data collection?
Yeah, the temperatures we're seeing are definitely on the high end of anything looking back in time. And what's interesting about when I looked at the gray whale story in general, it makes me sad that Atlantic gray whales went extinct. But what's interesting, and sometimes you don't hear about the good aspects, not climate change isn't, I'm not saying global warming is good or bad, but something that's interesting is that as the Earth gets warmer, it's making it more likely that gray whales could move from the Pacific back into the Atlantic. So although it's a sad story, I sort of have hope for Atlantic gray whales that they could repopulate the Atlantic Ocean coming from the Pacific. But then there's a whole nother shore of issues with shipping lanes and whether or not the habitat is suitable anymore. But as far as climate records go, we are in a very warm stage, and it could allow for exchange of populations that have been isolated for a very long time.
I think that may segue to whether you have any ideas as to what might cause the species to speciate on opposite sides of the equator.
Yeah, so there's been a lot of climate scientists and other people looking at speciation in dolphins that have found it seems like there's a couple factors, and one factor would be a waterway closing, like in Panama, or if you actually have currents changing, and or potentially a hypothesis could be you have a whale that lives along the equator, but as the equator warms up, it moves to the northern and southern hemisphere to get to a colder environment. And just over time, that as that population or ancestor was separated, it just speciated. There were fewer interactions, and then maybe they never moved back to the equator. But so it's sort of large scale climate fluctuations like glaciers blocking landways or landways opening up in marine mammals, I would say, are some of the major speciation drivers.
How long does it take to run a genome comparison to see variations between possible different species? And what level of computing power do you need? And anyone else who has questions, if you joined us late, please use the chat and just type them in there.
So that's a great question. It's not something you can do on your MacBook or your computer. I have tried to look at a genome on my computer in a software and it crashed. So what I do is I'm part of a supercomputer cluster in Denmark, and a lot of agencies or universities have kind of a high performance computing system, and I will at any given point request maybe 10 cores or 20 or 30 cores of the supercomputer, and I will let a genome comparison run for anywhere between I have had something run for 14 days. So it's kind of a trade-off of requesting enough resources to get the job done in a timely manner, but not to request too much that you get put at the back of a queue behind people who are submitting less demanding jobs. But I would say the average job or genome comparison takes 3 days using anywhere between 10 to 30 cores of a computing supercomputer.
Morgan, if I may ask, just out of curiosity, where do you do your sequencing work? Is that being done locally, maybe in a core lab, or do you send it to a sequencing service or company?
Yeah, that's a great question. So the University of Copenhagen has, at the university, there's a kind of a national sequencing center. So a lot of sequencing in Denmark or Europe gets sent there, but the sequences I'm specifically working on right now were sequenced in New York at the Rockefeller Institute.
Okay.
They are part of, there's a vertebrate genomes project. They are part of a larger project to try to generate genomes for all vertebrates. So it kind of depends. There's a sequencing center in China called BGI, and people send samples there. I think it can come down to cost and how many samples you have. So it just depends really. But like all of the environmental DNA work that was conducted in the lab, that was all sequenced in Copenhagen.
Okay. And what's a typical turnaround time when you submit a sample until you get some data back?
Oh, less than a month. I would say 2 weeks would probably be average. Yeah.
Yeah. Well, that's pretty amazing, for the probably number of base pairs.
Yeah.
Okay, great. Yeah, are there any more questions? Susan, are you monitoring or?
Yep, I am. So there's one more sciencey one, and then I have some more that are more career advicey ones. So does acidification of the ocean due to carbon dioxide impact whales? Or have you looked at that at all?
It's not something that I'm that familiar with. If it was, I would say if it's related to the food sources, if whales can no longer feed on their prey because of changes in those sorts of conditions, then I imagine it would. Specifically, otherwise, of the species I work with, I'm not very familiar with ocean acidification, if they have an impact on the distribution or health of whales right now.
Okay. So let me jump to some student questions about think back to when you were a Cohasset High School student. We want recommendations for people who want to be a marine biologist, study marine biology. And they also want to know what you're planning to do after your PhD.
Okay, so recommendations for students who want to study marine biology. I would say one thing that I would work on as early as I can are sort of the technical computing skills. So I came into marine biology because I love the ocean. I love marine mammals. I really love coral reefs as well. And sort of those were the things that drew me into the field. But the reality of it is I do spend most of my time now doing bioinformatics, writing little scripts, submitting jobs to clusters, using existing software. And the earlier you can become familiar with Python, Bash, Unix, just command line, I think the easier it will be for you to adapt to the required skills later on in the field. So that would be recommendation, is if you can get into a introduction to programming or computing class, definitely do that. Otherwise, I think just follow what you're passionate about. As you can see from my path, it's not like I studied marine biology for my bachelor's and master's and then went into the PhD, but I kind of navigated my way through. I even avoided genetics in my undergrad because it scared me, and I wasn't interested in it until it started to be applied to whales. And then I played some catch up, but I just want to point out that you don't have to have it all figured out from the beginning. So, but I think the technical stuff, if it doesn't help you in marine biology, will help you if you have a career change anyway. And the second question was what I'm going to do after the PhD. And I will probably apply for a postdoc and try to continue some of the work I've been working on. And a postdoc is just, once you're kind of established yourself as by finishing the PhD, you're sort of in the in-between stage where you can do things more independently and run your own projects, but you still need the support of an existing lab. And so I'll likely apply for funding in the next, probably in 6 months, for postdoc funding to try to complete a spin-off project from what I'm currently working on, or maybe I'll try to undertake something a little bit different, but within the field of marine mammal genomics.
Morgan, I have a question for you. I spent most of my life at sea, and I've been a great observer of wildlife and sea life. And I noticed that particularly from the equator north and the equator south, you see the same species of birds are, as far as visibility they are. At the further south you go, the further north you go, you see the same things occurring as you go along. I don't know how that affects whales or other fishes because you can't very well see them too well. But I'm just wondering with the southern right whales, I read a pretty good story about somebody did some research down there in the Patagonia about them several years ago. But if it was possible to transport some of the southern right whales up here to the north, would they be genetically similar enough that it could help reinforce the population before these fellows up here go extinct?
Yeah, so that's a really interesting question. And I think the first thing you would have to think about is if the northern Atlantic right whale and the southern right whale, if they've been separated for so long that even if they were in the same population, whether or not they would breed. So that would be one thing. I have a feeling they might still, depending, I'd have to look on their divergence date. But another consideration is that one thing people are noticing who studied endangered or isolated populations is that sometimes a population that's so small has kind of weeded out all the bad eggs and mutations. And if you bring in a new genetic pool, say from a distantly related or distant population or even a closely related species, what could happen is you could introduce some new mutations that would be bad for the gene pool, and that could result in further population collapse. So it's risky, but people have done reintroductions like the Isle Royale wolves, and it's a nice thing to think about. But practically speaking, I don't think it would happen with the southern right whale moving into the northern hemisphere. But I like that question, and it's something I will continue to think about. And it's I wonder why, to begin with, what drove the southern and northern right whales apart, and how genetically, how distant they are or similar. So I like that question.
Thanks.
I wonder whether the oil and gas explorations, when they use the underwater explosions, have something to do with where whales will go now. That was something that we looked at in a movie called The Sonic Seas.
Yeah, for beaked whales, that is a big consideration in general, is because oftentimes after even naval exercises or anything with intense sonar, there can be mass strandings of beaked whales, and it's a bit unusual to find otherwise species that are relatively cryptic, that they're all of a sudden stranding in large numbers. And that's a research field that people are looking at, is what are the effects of acoustic noise and also sort of underwater oil and gas exploration, and what effect that has on population health of beaked whales. Because if an island chain only has a population of 100 and 25 of them wash up every time there's a naval exercise there, it could really put the population in danger. But that's an ongoing research question that I personally don't work in right now, at least.
Yeah. So I think we were kind of hoping that you were going to say after your PhD, you'd come back and be at CSCR and take us out on deep sea whale explorations and teach us about genomics, because you just did such a great job of it. But I do have an excellent question from Ashley. She says, thinking back to your time at CSCR, what skills did you gain then, back when you were
Yeah.
that you still use in your research today?
So I think back then, one thing that stands out to me was the lab notebook. And I try to keep everything in my life in general in my head. And I don't have a calendar. I'm not very good at writing things down. But one thing you have to do in science is take notes on what you do. And I remember it every day when we would come back from the field. I'd want to grab my stuff and bike off to Atlantic Bagel or whatever and go to the beach, but we would always have to log in our lab notebooks what we did. And that is kind of carried through today, because if I'm writing a script and I need to go work on a different project and then work on a presentation, and then maybe I have a course, if I go back in a month from now, it's scary, but I might look at my data and think, what was I even working on last month? But if I keep really detailed notes, I can say, oh yeah, I was running this script and these were the results, and this is where I was going to take it in this direction. So I think sort of documenting what you're doing is something that even though I'm continually trying to improve on it, it was one of the things that was pointed out in CSCR that one should be doing in science.
That might put Mr. Buckley on that Mount Rushmore of Cohasset High School teachers that's trending now on Facebook. So are you still doing paper, or do you use an electronic system for your notes?
So I usually use an electronic system. And so I keep a folder of directories that kind of say analysis by species. And then if I start a new project, I'll create kind of a shell. It'll say readme.sh, like shell. And then in there, I'll have hashtags with what I'm doing, where the sample came from, where I downloaded the data. If I have any information about where or who did the sequencing or what lab techniques were used, I'll try to keep all of that in one place. If I do have paper, it's for kind of sometimes on literature review. If there's a fact I really want to remember, I'll write that down in my notebook. But I try to keep everything online and in the cloud, so I can find it and so that it's backed up as well.
I just noticed that when you said the thing about that you recommend that people get the technical computing skills. And I think, like you said, a lot of students who are interested in marine biology are more interested in animals to start out with. But we found the same thing even with our eelgrass group. So we're snorkeling, we're swimming in the eelgrass. It's wonderful. It's really cool. And we found over this past year that we collected so much data that we resorted to Python to help process it. And when you realize that the computing skills actually just make the tedious part of your data collection or your data analysis easier, then it's a little bit more of an incentive to go learn them. So we've used Python, and then we've also decided that we could go with Google's teachable machine and we're doing image analysis. So we're having computers do some of the work that we would otherwise do. So I think that what he said is valid, even at the high school level. We're finding that to be true too. But it's not dreadful. It's fun. I don't know if Beck Lavash is on here, but he's been really great about it. Any other questions? Let's see. I think you covered my regular questions, which are like, what's the coolest part of this? Your earliest taste of science and all that. Your favorite part of your CSCR experience?
I think just being out in the field and being with friends. I mean, I was in a group of people I would want to be hanging out with anyway, a lot of friends, and we were out in the field and learning and being in the marine environment. So I think for me, that was just taking advantage of what Cohasset has to offer in the summer, and being out in the field was probably, one of the things I really enjoyed about CSCR.
Excellent. Thank you so much. Karsten, I'll let you wrap, and then I'll mention, let me just mention a quick announcement, and then I'll let Karsten wrap up because he's the host of the lecture series at CSCR. So upcoming, looking forward, we'll have more lectures coming up. We always welcome alumni back. That's really exciting and rewarding for us. I think Jacqueline had said that she was really honored to be part of your presentation. I would love to meet her too. Looking forward, we also have CSCR's annual State of the Harbor coming up, and we expect that to be on April 1st. If you're a student and you want to get involved in that, you should be getting involved and reach out to your project supervisor. If you haven't been involved in a project, you can still reach out to us.
Just winding up right now.
For you.
Oh, good. All right.
And then, and we're also looking for sponsors for that event. So if you're not a student, but you think it's great that we get students out sharing their research with stakeholders in the community, please consider sponsoring the State of the Harbor. Okay, I'll pass it back. It's lovely to see everybody. I'll pass it back to Karsten.
Great. Thank you, Susan. Well, Morgan, thank you very much. That was an awesome presentation, and also catching up what you have been really doing. I'm very, very impressed. And also thank you for bringing your whole family here online. And it's also good to see a couple of friends here from town, which doesn't happen so often anymore. The Leffel family, hi there. I saw Giddy and her husband. And anyway, so we're trying to keep this going on a monthly basis. And I think February 25th is the date of the next virtual presentation by the Norman brothers. We'll get into more details. I just have the date at this point, but we'll keep on sharing details about this. Check back, and we'll probably keep this format going until Mr. Fauci allows us to do something else again. Anyway, thank you again, Morgan. That was wonderful what you did here for us. You enlightened us with also these videos, which you probably took yourself and edited yourself, right?
Yeah.
Yeah, that's awesome. And I'm sure you're certainly inspired a couple of people here. Great. So I don't know, Susan, Jack, you want to do the final mouse click to X us all out? Anybody else want to share something or say something?
I just wanted to say again, thank you to everyone who tuned in to listen. It's nice to see so many familiar people and from people at various points in almost all of those phases of my career. And there's people from Australia, Phil's listening in from California, people from my undergrad, and people from CSCR. So just thank you all for tuning in and showing you're interested.
Okay.
And you have some nice compliments.
Can I just give a quick shout out to Morgan and say thank you. That was awesome, Morgan. And thank you for all the people that Morgan just mentioned who have been working with Morgan and supporting him. And it's great to see so many people, all the familiar faces, but great to see some former students here. And Dom, great to see you, and Ashley and Christine and Lily, and forgive me if I miss you, Colleen Richardson. And so lots of great memories with some of the names up on the screen, and it's great to see so many people together. see Levente's here too. And so thank you all. That was great.
And if you haven't connected with CSCR, please make sure that you have gone to our website and signed up for our emails. That's where you'll hear about the next lecture. So that's C for Cohasset and then cscr.org on the website. And we'll just leave this open because one of the things that we've loved about our lecture series is that people get a chance to talk with each other and ask questions and interact. We normally have, snacks and stuff afterwards. So we'll leave this open for a bit until people want to head home.
Okay.
You can unmute yourselves. And
Thanks, everybody.
Thank you, Morgan.
Thank you. Yeah, that was terrific.
Thanks, everyone.
Learned a lot.
Was that Emily Shea back sticking her head in earlier? Who was that? Was that
I was.
Julie?
Emily, you should come and say hi. It was. All generations here.
That's awesome.
Morgan, it doesn't sound like you're getting out into Copenhagen so much.
No, no, I've been
Studying.
in my office a lot, but now I'm actually at home in Cohasset, and I came back for.
When are you going back?
So I was going to go back on Sunday, but my supervisor suggested it would be okay if I stayed to watch the Super Bowl. So I'm thinking I'll continue working remote until probably the 10th or 9th or 8th, and then fly back. But everything is pretty locked down now. The offices are officially closed, and the laboratories are still open. But I'm just doing data analysis with the supercomputer, which I can access through VPN in Cohasset anyway. So yeah, it's nice to spend some extra time at home and of course be here in Cohasset.
Nice you were here. Thank you so much.
Yeah, thank you for tuning in.
Okay.
our CSCR computer has a program on it that that means that when it's on, it helps process other people's data.
Is it part of Galaxy?
No, I can't remember what it's called. My brother put it on there, but as it's running, it like lends its capacity to other researchers.
That's really great. And it's really good to hear you guys are incorporating all the technical skills now. And I think it's not until you need to solve a question you're interested in using those technical skills that you think they're important to your life. Just like I wasn't interested in genetics until I learned that you could answer questions about whale ecology and biology. And if you need those technical skills to get to that answer, all of a sudden you really like Python or Unix or Java or whatever you're using.
Hey, Morgan, that raises a question because I was going to kiddingly say, wow, when did you get so smart? And because I'm deeply impressed by your knowledge and how well you did the presentation. But my question is, did you ever have any have you had doubtful moments like, can I do this? Is this the right field, or is this too challenging for me? Any doubts along the way?
Yeah, definitely. I mean, all the time. I think there's always somebody else who's smarter or has a better idea or who has more years of experience than you. And I think you have to remind yourself that everyone's kind of just sort of figuring it out. People come off more confident than others, but I think at the end of the day, people in the field or working in genomics and genetics have similar. I mean, it can be difficult to grasp, but if I talk to other students kind of to gauge where I'm at compared to them, they have the same questions, or I might think, oh, that's interesting. They didn't know that, but I do know that, and not coming from a genetics background. So sometimes I feel like I have played catch up in a bit because I wasn't as into genetics in undergrad. But I think throwing enough time at it has put me on a playing field where I'm comfortable speaking with other students who do genetics. Of course, it can become intimidating when you're speaking to the leaders in the field and they're asking you about the specific allele frequency or something very specific in genetics. But I think, I'm slowly just building my knowledge base and then building off of that. But yeah, for sure, I have doubts sometimes.
No, that's awesome. Thanks.
And it looks like you align yourself with good people. Like
Yeah.
there's an opportunity and you're like, yes.
I've had a lot of people who have been really supportive, or like people who have, people who are running laboratories or whole schools sit down with me and look at a mitochondrial genome and zoom in and say, hey, that's the gene. We've got 13 of these. And just people who have taken the time to explain things or given me the chance or the environment to learn it myself. So I think it's kind of a combination of being willing, being driven, inquisitive, but then having those breaks or those opportunities where you're given a chance.
Okay.
All right.
Any further questions here? It's getting close to dinner time for everyone, I think. Yeah. So good. Thanks again, Morgan. And Jack or Susan, can you close us down here?
I want to give you a sample of water from off of Cohasset now and find out how many species you can detect. That's unbelievable. It is totally mind blowing.
It would be really
If
cool.
you are around this summer, you definitely have to do a gig appearance and run us through some lab stuff.
Yeah, definitely. Hopefully I'll be coming back depending on all of this. But yeah, I intend to come back at least.
Great.
So.
Yeah, thanks again, Morgan. That was super. It really was awesome. And Karsten, always thanks for putting these things together. And Susan, you're the hostess extraordinaire, MC extraordinaire. So thanks again for playing that role.
Well, thank you guys. Have a good evening. And
McCarthy's and Habers.
And, thanks to everyone who dialed in here.
We'll see you guys later.
Just up, and there's more to come.
Thanks, Gita.
All right.
All righty. Thank you. Bye-bye.
Great, thank you so much. Like people who.
I love this journal comment before you hang up. Ah! I get on my phone, I text it to you and say, oh, he just said journal, it's portfolio.
You didn't just say journals, he really went into it. Yeah. Did you pay him to say that?
Anyways, that was all good. All right, great job.
Yeah. Hey, is Levente still on there?
What's that?
Is Levente still on there? Hello? Hi, Levente. Maybe not.
Don't know.
Just
all right. We'll see you tomorrow.
Yeah. No,
Saturday morning.
Yeah, 9:30.
Yeah.
And then, yeah.
All right.
And let's hit the off recording. You're going to get a really bulky file from Zoom.
And then we can exchange our usual 100 emails tomorrow.
Yeah.
No, it was.
Oh, so tomorrow morning,